We are live.
Hello, everybody. Welcome to this session where we'll be sharing with you some of the views and approaches that we take with regards to transparency and reproducibility in the British Journal of Pharmacology. I'm here with two of my fellow senior editors, Professor Mauro Teixeira, whom all of you know, and probably I don't need to introduce, but just to remind you all, he's Professor of Immunology at the Department of Biochemistry and Immunology at the University Federal de Minas Gerais. I hope I said that right, Mauro. He's also the Latin American Senior Editor for BJP. He's going to be speaking to you about our guidance on natural products in due course. My other colleague that's here is Professor Claire Stanford. She's Emeritus Professor of Neuropharmacology in the Faculty of Life Sciences at University College London. She is also a senior editor at the British Journal of Pharmacology with a focus on neuro and psychopharmacology. Claire has a lot of experience with NC3Rs, so the 3Rs and animal experimentation. She has played a major role in the National Animal Science Committee and is a lesser trustee organisations within the UK. And she's going to be speaking a little bit later, in fact, after I've introduced the British Journal of Pharmacology about sex and as an experimental variable. So without further ado, let me tell you a little bit about BJP. I'm just going to share my slides. Hopefully this is all going to work well. I hope you can see my slides. Mauro, Claire, could you tell me if you can?
Yes, we can.
Yes.
Great, wonderful. Okay, so actually, this webinar is part of a series of webinars that we've been doing to celebrate our 75th anniversary. That's essentially the British Journal of Pharmacology has been going for a very, very long time. The first editor-in-chief of the journal is this chap here, Sir John Gadam. I'm sure as pharmacologists, all of you are very familiar with Sir John. He discovered substance P, and actually he was one of the key individuals at the British Pharmacological Society who supported the establishment of the British Journal of Pharmacology. 75 years later, this is me. You see me. I think this is probably a photo that makes me look a little bit better than I do look today, but I'm the first female editor of the British Journal of Pharmacology. I have some similar interests to those that Sir John Gadam had. I'm interested in sensory neurons. I describe myself as a cardiovascular translational pharmacologist, and tomorrow I'll be speaking in a keynote lecture about my interest in sex differences. But I've put this photo up because I think actually they're really quite good in reflecting an important feature of the journal. When the journal was incepted, it was largely a journal that was supporting the publication of British pharmacological research. Today, we're a very diverse international journal supporting the publication of research from the global pharmacology research community. And this is reflected by the membership of our editorial board. As I mentioned, Mauro is our Latin American senior editor, and we specifically, I specifically, recruited, is that still showing? Are you still seeing my slides?
Yes.
I specifically recruited Mauro because I realized how important pharmacology research in Latin America was. And it was important for us to reach out to your community and to engage you more in publishing your excellent pharmacology in our journal. We have a number of experts who have focuses in statistics, design and analysis, clinical pharmacology, redox chemistry, and medicinal chemistry and drug discovery. And all of these individuals support authors who submit their work to us. The true diversity of our editorial board and the journal is reflected by the fact that we have editors that come from 22 different countries at the moment, and 30% of the editorial board is female. The span of the international representation of our editorial board exactly well replicates the span of the international origins of the individuals who publish it in our journal. Oh, it's not changing slide. Whoops. Okay. This is just a selection, an image that actually depicts the kind of papers, the broad remit area of the journal in terms of our publications. We're, as I said, a broad remit journal. So we publish pharmacology in all specialties, ranging from inflammation to neuropharmacology, to cardiovascular pharmacology, focusing on the genitourinary tract. And of course, all of the publications in the journal have to advance, provide significant advance in mechanisms of action or the description and discovery of novel therapeutics at new targets. This summarizes the kind of work that we publish in the journal. We publish, the main job of the journal is to publish original research, but we also publish state-of-the-art and authoritative reviews. These can be either mini reviews in hot topics that are emerging, but also larger reviews that provide an update on targets or novel therapeutics. We also have a new fast track mode that allows the very rapid publication of really important findings that really need to get out to the research community as fast as possible. As I said, we're a broad remit journal. We publish in all fields of molecular, in vivo, and translational pharmacology, but the work must have a direct bearing on the effects, mechanisms, or uses of drugs, or the development of new drug targets. Very important is that we don't publish work on biological extracts of unknown chemical composition. However, if you have actually identified the active component or components within your extract, then that work may be suitable for the British Journal of Pharmacology. And I'll leave Professor Teixeira to give you more information on that subject. Our primary aim at the British Journal of Pharmacology is to publish as much excellent pharmacology as we can. We're not a journal that is seeking to restrict the number of publications that we publish annually. We want to publish as much good work as we possibly can. In addition to being a mouthpiece for the international pharmacology community, what we also do at BJP is share best practice with the global pharmacology research community. And we do this by publishing guidance documents on key issues that are pertinent for pharmacology research today. Important to us is that we've had a number of initiatives, and these are listed on this slide. First of all, nomenclature of drug targets is very important for the pharmacology community, and in support of a unified nomenclature approach, we publish something called the Concise Guide to Pharmacology, which is a snapshot of the IUPHAR Guide to Pharmacology database. For those of you that haven't looked at the Guide to Pharmacology database, I urge you to go there. It's an absolutely excellent resource that provides key information on all known pharmacological targets, provides information regarding the best drugs that might be used to interrogate that target, but also details on perhaps clinical development of those targets and medicines that are specifically targeting that receptor, enzyme, transporter, channel. As I said, we also publish guidance to enhance transparency and rigor of preclinical research. And we have focused our efforts on key areas that we feel are very important and pertinent for pharmacology research at the moment. We publish experimental design and analysis guidelines. We've provided guidance to authors, editors, senior editors, reviewers on the information that's required for replication of animal experimentation. And this is based upon the ARRIVE 2.0 guidelines. We're going to hear about sex as an experimental variable and the guidance that we have most recently published from Professor Stanford. Natural product research is a major area of publication in the journal. And so we've provided clear guidance on what our expectations are of that research for it to be publishable in BJP. And we'll hear, as I said, from Ambrita Shera. There's also guidance on immunoblotting and immunohistochemistry, on the processes for making data available. And very recently, we published some guidance and a position statement on where the journal stands with regards to preprint citation. One of the key things that we've done in the journal is that we want to make sure that others can replicate the findings that are published in our journal. And one of the ways in which we can do that is to provide clear identification of all the resources, the reagents that are used by the scientists that publish in BJP. And so we've collaborated with some colleagues in San Diego, at San Diego University, with regards to the SciScore initiative and the database that they've established of the Resource Identification Initiative. This actually provides ID numbers to individual reagents and resources, and we cite those numbers in our manuscript so that others can get the exact same reagents for any replication studies or follow-on studies that might be done. And then we share all of these efforts of ours are in a bid to provide that endorse and support the Open Science Initiative. And as part of the Open Science Framework, there are guidelines on that have been set essentially by the Open Science Initiative that allow a clear indication of how good we are at supporting the open science agenda. They're called the top guidelines. The BJP does very well in these top guidelines, and we share the information, our ranking and rating within the top guidelines on our website. So please go to our website and take a look. The editorials that we publish on each of these important areas are freely available. But what we've also done is created checklists that actually can be downloaded and used by researchers when designing their experimental work. So they're available not just as a tick box exercise to make sure you've fulfilled all the requirements for submitting your paper to BJP, but actually we've created what we feel are resources that can help you when you're at the design stage for your experimental work, providing advice and guidance on what might be the best way to approach your research question. And these are the DOIs for each of the key articles on guidance that we've published in the journal. Just to end, I just wanted to do a little bit of a plug for the World Congress of Pharmacology that's happening in 2023. I'm the Secretary General of the meeting. This is David Webb here. He's the president of the meeting. You've got the images here of all our fantastic keynote speakers, a number of Nobel Prize winners, ranging from COVID to Neuropharm to iPSC research. It's going to be a fantastic meeting. It's going to be an in-person meeting in July 2023. And I really hope that myself and David can welcome you there in Scotland in July of 2023. Okay, so now I'm going to hand over, I'll stop sharing, hand over to my colleague, Professor Claire Stanford, who's going to tell you a little bit about our guidance on considering sex as an experimental variable for your experimental work.
Thank you, Ambrita. I've just realised that all the questions in the chat line are in Portuguese. I suppose we should have worked that out. Mara will have to translate. But anyway, thank you for that introduction. I'm going to be talking about guidelines on design analysis and also, as Amrita mentioned, our policy on dealing with sex differences. Now, the journal has published a couple of editorials in the past, and I really urge people to read those because they explain features in the instructions to authors. They explain why we ask for particular elements which must be satisfied, which affect the experimental design and data analysis. And many authors do appear to submit manuscripts where many of these factors aren't included. And that's
Professor Claire, I'm so sorry to interrupt you, but you are sharing your screen. That is not your presentation. If you want to open up your presentation on the screen you're sharing, I can add it here on the stream. We are seeing your calendar, if that helps you over there.
Oh, right.
Okay, no problem.
Okay. I don't see. Is that better?
It's not showing for me yet, but if you can try to press the share button again after opening, like you were doing it. Not seeing, Professor, if you wanna. Okay, right now. Yes. Yes, it is. Can you just press the presentation mode, please?
Okay. Okay. Thank you. So these
You're
are the
very welcome.
Thank you. These are the editorials I was referring to.
It's not on presentation mode yet, Professor. If you can just click it here, like to present. It's we are seeing your PPT.
So all I've got is stop sharing.
No, on the PPT, can you press the button on the bottom that is like a monitor? I don't know how, if we are not seeing as slideshow. We are only seeing like the PowerPoint, but not on presentation mode. You are seeing as you're presenting?
Well, all I can see is hide and stop sharing.
Okay.
And it worked before.
Yes.
Doing what I've just done.
Okay. So I think we can keep going if that's okay with you. I mean, we can see the slides. We can, no problem.
Is that, I don't know what people are seeing, but
we are seeing your PowerPoint, but like if you are editing the PowerPoint and not presenting, you know, as in slideshow.
Okay, well, we'll carry on with the PowerPoint, I suppose. Okay, so these are the editorials that I was talking about, and they explain what's in the instructions to authors, and we really do require certain features that must be included in the manuscript for it to be considered for publication. So if those factors are actually missing from the manuscript, many don't then go on for peer review because it's really not possible to evaluate the work without that information. And those factors include things such as the experimental groups should all be equal size by design, which confirms that the study was fully randomized. The study really needs to be confirmed that it was blinded to, and ideally in the data analysis step as well, not just while carrying out the experiments. We also require all experimental groups to comprise samples of n equals 5 at least, so that when you're doing two group comparisons, you can use valid parametric statistics. With n smaller than 5, that's not really feasible. Also, for valid parametric statistics, we need to be reassured that the sample variances are the same. And if we're doing repeated measures analyses, then confirmation that the variance covariance matrix should be spherical. So you have, that involves the sphericity test. And although we don't encourage exclusion of data, sometimes that really is inevitable for justifiable reasons, but we need to know all those details. What were the criteria? What was the test used for excluding data? And also to provide a full audit of the numbers that were missing from the original data set. And of course, also a single p-value throughout. But the guidelines that I've been most closely involved in are those on how to deal with sex as an experimental variable. And reading these guidelines, you'll see there are lots of examples of where the sex of the subject really makes a very big difference to experimental outcome. There are lots of examples of where physiological systems can differ, where pathology, illness differs, or vulnerability to illness differs between the sexes, and also examples of where drug pharmacodynamics and the drug responses can differ between the two sexes. So that provides a background to why we've developed a policy on sex as an experimental variable. Now, many groups describe sex as a biological variable, and we take that as an absolute given. And what we want to do
Professor Claire, I'm sorry, I'm the one bothering you again, but we are not seeing the slides. It's not changing. It's showing the same slide.
It's changing on my screen.
Yes, if you can try to share your entire screen, maybe you just shared the window, and that's why we're not seeing the same thing as you are.
Okay.
So I would appreciate if you could stop sharing. And okay, right now it changed. It's
okay,
on the,
so we'll have to do it on, we'll have to do it on the slide show rather than full screen, so you can see this.
Yeah,
okay.
yeah, we are still seeing your PowerPoint, but we can see that you changed the slide now. Okay.
Okay. So the examples of where sex makes a difference to the experimental outcome, which is why we want to think of it as an experimental variable, which has to be incorporated into the experimental design, rather than simply as an extraneous biological variable. And that follows policies from organisations like NIH and also UK research councils, where they really are expecting people to provide information on research outcomes in both sexes. And that the reason for that follows several key concerns. And one is that the majority of preclinical research has a sex bias. It's concentrated almost entirely on males. And the reason for that is historical, that many decades ago, there's many publications suggesting that the responses to physiological challenges and behavioral challenges in females in rodents was far more variable than in males, and that encouraged people, therefore, to study males because they get more consistent results. I have to say that recent studies that have carried out large-scale meta-analyses have led to some considerable doubt about that, that people really are failing to see any higher variability in females than in males. But the other reason, of course, followed the thalidomide catastrophe, which led to clinical trials being under intense pressure to avoid recruiting women, particularly of childbearing age, and carry out early studies in males too. So from those two directions, it's easy to see why a huge tranche of scientific literature is based on studies only on males, but that is changing for important reasons. And one of those reasons is, that I've already explained, and as you'll see in the guidelines, that there are important differences in biological and molecular mechanisms of action and the actions of drugs in males and females. And also, and this is really what bumped NIH into action, is that between 1997 and 2000, 10 medications were withdrawn from the clinic. FDA withdrew the license for marketing, and of those 10 drugs, 8 presented clear greater health risks for women than men. And they include some well-known drugs like tafenidine, the antihistamine, and others used routinely for gastrointestinal disorders and so forth. And finally, from the preclinical point of view, there are ethical concerns which are obvious with overbreeding of female animals. Now, this applies particularly to genetically altered animals, but if studies are only carrying out, only using males, then hitherto the female animals have been regarded as surplus requirements, and they've undergone euthanasia. So they've lived and they've bred and lived for absolutely no purpose at all, when we can learn a very great deal by using them in our experiments as well. So the policy now from the BJP is to require sex to be considered as a factor of interest in the experimental design. So all experiments in vitro, in vivo, and ex vivo should include both sexes whenever possible. And we require also the use of one or both sexes to be reported in the manuscript. And if authors don't use both sexes, then there should be a statement in the discussion to acknowledge that the findings reported in the study might not apply to the sex that hasn't been studied in that experiment. And ideally, we would like the sex that was studied to be highlighted in the title of the manuscript as well. Now, obviously, there are examples of where you can only study one sex, such as when you're looking at uterine tissue or vas deferens, for instance. But with those exceptions, we would like to see a scientific justification for studying only one sex. And very often we see defense of that with authors saying, well, we don't know whether our experiment will work in females because it's only been done in males. And so we're going to carry on using it in males and might get round to studying females eventually. And we want to encourage change of mindset, where the starting point is to study both sexes. And then if there is a sex difference, to interrogate that in more detail and carry on using only the sex where the experiment worked, knowing that it's a special case rather than a general rule. And that also applies to immortalized cell lines like halal cells, for instance, and also, of course, tissue derived from a sex organ. But another justification we often see in manuscripts is authors saying, well, we want to comply with reduction in the 3 R's, and if we study both males and females, then we'll need to use many more animals. We'll have to use twice the number of animals in the experiments, and our ethical approval body won't allow us to do that. Now, I want to convince you that that is absolutely not the case. It doesn't have to be so. And this is why, that if you incorporate sex into the experimental design, so you have a factorial design where you're looking at the effects of sex as one factor and the effect of the drug as another. And being a pharmacological journal, of course, we like to see dose response curves, not single dose studies. So we would like a low, intermediate, and high dose with vehicle. So we then have 4 groups. Now, in a properly powered study, say we would normally require 10 animals to reach a safe conclusion. Now, in a single sex study, that would be 10 males. But if we use both sexes, then you can use 5 males and 5 females. And that still gives us an N of 10. And it gives us an N of 20 for to interrogate our drug response. So that would leave us with plenty of animals to look for a main effect of sex, a main effect of the drug, but also has that powerful factor where we can look to see whether the effect of the drug depends on sex, because the 2-factor ANOVA will tell us that there's been an interaction between the sex and the drug factors. And with N of 5 in each cell, then we can still go on and do post hoc comparisons looking for a sex difference at particular doses. So it's really important to realize that it's not an infringement of the reduction, and that in an adequately powered experiment, we can look at both sexes perfectly feasibly. So that's the closing message, just to encourage you all to use both males and females in your experiments, whether it's whole animals or cells in culture. So thank you for listening, and I'm now going to hand over to Maro.
Just before we start with Maro's presentation, just to let you all know that we will take any questions that you have at the end. All three of us will come back. If you could post your questions, preferably in English, but if it's in Portuguese, that's fine too, in the comments. I think you can access that from YouTube. Is that correct, Maro? So if you post your comments there, we'll take all of them together at the end. Please ask any questions about the journal or about any of the issues that are brought up by the presentation. So now I'll hand over to Professor Teixeira to tell you about natural products.
Okay, thank you, Ambrita. It's nice to be here. I think my screen is now popping up. Okay, so I will briefly discuss some of our ideas about natural products and the stance the journal takes on it. And as Ambrita said at the very beginning, you know, BJP is really interested, oh, come on, is interested in excellent pharmacology, and it will publish, you know, any work with excellent pharmacology. In the context of natural products, it is very important to realize from the outset that we do not publish work on the actions of biological extracts of a known chemical composition or a known concentration. So we are really talking about molecules derived from natural products, and especially molecules with a known mechanism of action. And I will actually detail that a little bit more. So there have been a lot of experiments and studies on natural products published in BJP. We've published recently our guidelines for natural products, and this can be found here. It's easy enough to find in the journal as well. And as put here, you know, we are interested in novel drug targets, elucidate the mode of action, discover lead compounds, and ideas that offer potential for natural product repositioning. So there are a lot of papers published there, and it's simple enough to follow these guidelines that will make, you know, the process of submission and acceptance much easier. Right, so these are the 10-item checklists that are necessary, and I will sort of highlight the major ones. So we are obviously interested in molecules derived from natural products, and we are interested in at least knowing the precise mechanism of action of an indication of it, you know, a good indication of how that molecule works. So this is a very important point. If we are talking initial experiments actually started with a complex mixture and extract, we want to make sure that that goes all the way into the active constituent, you know, and preferably that, you know, if possible, to have a synthesized preparation, or at least a very purified preparation of that active constituent from the complex mixture. And there will always be example between, you know, below, but, you know, if you can focus on the top part here, we are really interested in this active constituent. It would be nice if we are really talking about the purification, that the purification process is given. Obviously, we want people to reproduce the work if possible, right? So adequate description of the extraction procedure or the synthesis of the material is needed so that we can actually chase that in much more detail. You know, it is important that the biological activity of the extract and the purified, constituent are the same. If we're talking that, you know, we started from a complex biological mixture that does something, that has an anti-inflammatory effect, we want to make sure that that activity seen in the herbal extract is also seen with the purified constituent, because then we can make inferences about that particular constituent being responsible for the biological activity of the extract. In many instances, and I'll say actually all instances, we want to see an appropriate vehicle control to, you know, to realize that what we're seeing is not really to do with the vehicle. In other words, those of us that work with extract, we know that we use a lot of organic solvents, sometimes vegetable oils, to purify, to add, to dilute our molecule. So it is very important that an appropriate vehicle control is added, as it is a positive control. So what are we comparing it with? What is that we want with our new drug? And it is always nice if it is an inflammatory model. It's always nice to check, for example, against dexamethasone, what does our compound do? So we're really talking about having a vehicle control, which will have whatever used to dilute your constituent, have a positive control, which sets the maximum inhibition, or it sets the example where we want to compare our things with, and then we can use our molecule of interest to compare with both, so that we have an idea of the effects of the vehicle, and we have an idea of where do we want to reach with our results. And obviously, we want that molecule to be pharmacologically relevant. And it's obviously difficult to say pharmacologically relevant, but obviously we are really talking about reasonable concentrations, so that the concentrations used in vitro are reasonable and are appropriate for pharmaceutical development. And there's no clear-cut limit for that. But obviously, if we're really talking about effects which are seen at concentrations higher than 30 to 50 micromolar, it's very difficult to develop that pharmaceutically. And you actually run into the possibility of actually having a lot of side effects. We want to make sure that those used in vivo is reasonable enough for the in vitro concentration. So an idea of the PK, of the pharmacokinetic of the drug, and what you can reach in vivo is also nice, because we want that in vivo dose to be translationally relevant. If in vivo experiments are carried out, route and time of the administration are appropriate for this in vivo translational relevance, and actually appropriate to reach adequate in vitro concentration. And if possible, also to assess the potential toxicity of the compound. It also adds to the pharmaceutical development, and it will also add to the relevance of the overall findings. I think these are the major points that I wanted to share with you. So I have to go back here and stop sharing so that we can actually discuss, this in more details. Thank you, Ambrita.
Thank you very much, Maro. So I think our colleagues are a little bit shy. I can't see comments at the moment. I hope that I've got the right part of the system. Maro, I don't know whether you can access the YouTube bit directly, but here I can't see any comments at the moment. So please ask your questions. If you prefer to write in Portuguese, then please go ahead. We have Mara here who can translate. So the question about sex as an experimental Portuguese. Then, I can hear myself. So the question about sex as an experimental variable, I think the key thing really are numbers. That's the cost of that, Claire, and that is something that actually plays a big part in many researchers deciding what experiment they're going to do. Funds are the key issue. And so you say that if your sample size says that you need 10 in your group to assess whether your effect, to assess a difference, if you want to, on top of that, really look and be powered to determine whether sex is having an influence on the outcome too, don't you have to do another sample size estimation for what you expect the sex difference to be too? So wouldn't you also, if you really want to be sure about sex differences, actually increase the N value?
Not necessarily. And with factorial analysis, the sample size will depend on what factor you're particularly interested in. So you may well come up with a different answer for the factor sex or the drug factor, in which case you'd have to choose the larger number. Because a power analysis will only deal with one factor at a time, and so you'd have to choose that number quite carefully. But actually, with more doses in the dose response curve, the matrix becomes more and more efficient. So the number of animals in each cell can go right down. It only becomes an issue if you want to then interrogate at a single dose. So if you want to do a 2-group comparison, then the view is, and the journal insists on having N of 5 at least, so that you can do a valid parametric 2-group comparison. But if you're only interested in whether sex or drug has an overall effect, you could have as few as 2 or 3 in each cell. But then you'd simply be left with knowing whether the drug had an effect or whether sex had an effect or whether it's an interaction. But it wouldn't tell you whereabouts in that dose response curve those effects actually appeared. So as a journal, we wouldn't recommend that.
And generally, for everyone who's listening, I mean, there's a big push by our funders that we start ensuring that sex is considered in our experimental design. So despite the fact that you might feel that there is a cost implication, actually, it's going to become an essential component of the research that we do going forward. It just simply isn't good enough anymore saying that I use males because it was easier and the standard deviation was less, because our research has to be relevant for both sexes and
absolutely.
population is important.
Absolutely, but I mean, what you often see is authors saying, well, I'm going to study males now because it fits in with everything I've done in the past, and I didn't want to do anything too radical, and we'll study females next. But that isn't satisfactory because, of course, it has to be randomized. You have to randomize males and females in the same experiment in order to make those comparisons.
Really important issue. So we have a question from Plinio Casarotto, who says that today there's a lot of in silico assays, for instance, docking analysis. What's the position of the journal of pure bioinformatics analysis of drug receptor interaction? Do you accept them? Well, in general, just in silico analysis is likely to be insufficient for acceptance in the journal. It depends really what you're showing, and it will depend on the review process. But we generally require some functional confirmation of the in silico observations. It's not just enough to run a screen and say it's likely that this compound will be binding to this receptor and therefore it's potentially a therapeutic. We need some biochemical confirmation that that actually is how it's working. So it would be important to do some translation, whether it's in vitro or in vivo. That depends upon the nature of the study, but it will be important to translate. How is it possible to randomize males and females in behavioral analysis? So this is again from Plinio. Plinio, just to say that we recognize sometimes that when you're doing experiments, actually randomizing the experiment, blinding the experiment at the time when you're doing it is a very difficult thing to do. And I think you're talking about blinding here. Randomization, actually, there are lots of available tools that allow you to do it. But what we say that if you want to truly have unbiased outcomes from your experimental data, that you can actually blind the analysis that you do. And there are ways in which one can do that. I think that might be answering your question, but I don't know, Claire, whether you think.
Well, I acknowledge this is a problem if the males and females have to be absolutely confined in the same environment at the same time. But we've studied males and females, and actually you randomize longitudinally. there's not a problem with that. And also, I think we mustn't lose sight of the fact that we'd then be looking at rodents in their real world. We don't normally find rodents living naturally with one sex in one environment, another sex in another environment. So by separating the sexes, we're already creating an artificial environment which could distort the findings. And it's fairly straightforward to build an experimental design where you can study both sexes and the behavior of them within an experiment without male-female interactions, the sort that I think you're worried about actually disrupting the results.
Okay, I hope that answers the question, Plinio. Myra, I think I pronounced that correctly. Have I done that, Myra? You can correct me if I'm wrong. Myra Beaker is thanking us for very well addressing the importance of accounting for sex differences. I wanted to just raise. Oh, please go, Myra. Myra, please.
I think there is a huge effort from the journal, not necessarily only about sex and natural products, but overall planning of results and overall planning of experiments. And I think this is the big push here. And it's not a push only for BJP because we are a pain. It's because there is a huge movement everywhere in the world for the likelihood that experiments are reproducible everywhere and they are meaningful. So that's why we are pushing a lot to have adequate planning. That means higher N numbers or adequate planned N numbers. Not the usual N equals 3. That's what I mean. That we push for randomization, that we push for blinding, that we push for both sexes, and that we push for adequate control when we're talking about natural product experiments. So I think this is a push worldwide, and in many instances BJP is actually ahead of that because we do believe this is necessary. And if you're really talking about drug development, this is really necessary. I think this is the best message that we can put forward today, correct, Ambrita? I think this is.
Yeah, I'd agree with that.
Yeah.
I'd agree with that. There are some most of the advice that we give in the guidance that we give in the journal is, all of the advice. There's a key aspect of the advice we give, and that it is pragmatic. We understand the limitations of the real world experimental environment. All of us are experimenters. And so whenever we create guidance, we're all thinking about our own ability to do the experiments according to the guidance. And we take into account funds, restrictions of funds, restrictions with the nature of experiments with regards to, for instance, blinding. And we try in our editorials to provide pragmatic solutions to help you address some of these issues. Solutions that can be applied by labs that are very well funded, and there are lots of individuals in the lab, but also for labs that might not have as many funds and as many people working in that environment, that the approaches that we propose work for both laboratories. Plinio is asking a really important question about registered reports. No, don't, please. You say that you promise it's your last question. That's it. You don't need to promise that. Please ask the questions. All of the questions you are asking are very important. So we're very open to accepting any protocols and studies that have been published in registered reports that won't bias against publication of your work in our journal. We recognize that for certain types of experiments, it's actually fairly simple to create a protocol that can be published in registered reports. But for other types of experiments, it might be more complicated. Certainly in the British Journal of Pharmacology, if you have a look at any of our papers, it's very rare that there's a single experiment reported in the paper. It's usual that there's been an iteration of experiments and it contains maybe 3 or 4 that lead to a final conclusion. Sometimes I think it's easier to perhaps publish a registered report of a protocol that comes at the very end of this research cycle. So we accept that sometimes it's doable, sometimes it isn't, but that doesn't bias against publication in the British Journal of Pharmacology.
Amrita, you may want to comment something about preprints as well, because that's a very important issue, and I think people are interested in that. Yeah.
The British Journal of Pharmacology has supported the concept of preprints since its inception, really in the biomedical sciences world. I mean, of all the sciences, we've been perhaps the slowest to adopt the preprint approach. So if you pre-printed your manuscript prior to submitting to BJP, that's absolutely fine. That won't stop your article from being published in BJP. However, what we have very recently made a statement about is that in publishing your manuscript, we will not we will not have citation of preprints in the bibliography. And this is largely because a preprint is essentially an unreviewed, unassessed piece of work. And actually, while it's important that there's a forum for the publication of preprints, we actually think it's also really important that experimental work is actually reviewed by the research community before the observations are accepted as de facto. So we support the preprint enterprise. It will not bias against you if you have preprinted your article. However, we will not, in general, unless there is an absolutely special reason that is accepted by the senior editor, we will not allow citation of preprints in the bibliography of any articles published in the journal. There are some further questions here regarding behavioral studies from Myra again. She says here at the end that she normally runs both males and females in separate experimental rooms on the same day under the same behavioral specifications. So that's an answer to Plinio. Although I recognize that sometimes that's a difficult thing to do. You have to have the availability of that space. I wonder, Claire, if you've got an additional comment there.
Just the fact that normally most housing units don't separate rodents by sex. So, they're used to living next to each other and smelling each other. So, they might find that the change in the olfactory cues in that environment actually quite even stressful. They realize that their environments change quite radically, and that could affect the experimental outcome. And you could argue that running the animals side by side is much more like what they're used to and much more likely to produce consistent results that aren't influenced by this sudden, rodents in particular rely on olfactory cues hugely. So if there's a big change in that, by suddenly finding they can't smell the boys anymore, that could affect experimental outcome quite profoundly. So
and actually, if you've included sex in your randomization plan, then actually you will account. I mean, the important thing is that you don't do all of the females on one day or in 1 week and all of the males in the next. If you actually use one of those there are loads of online tools freely available. You can actually do the randomization, or there is a really excellent program that is available from the NC3R's website. It's called the Experimental Design Assistant, EDA. Go to the NC3R's website, and you'll find instructions there that help you to actually design your experiment before you start.
That absolutely right, Ambrita, and I put in a big plug for the EDA. It's absolutely valuable, but at the moment it can only cope with single-sex studies. They are actually developing it further to allow both sexes as a factor in that software package. It's one of the rules, but at the moment it's not quite ready for that, I think.
So then you can actually just use one of the online randomization tools.
Yes.
If you go to those, you'll create a randomization table, and by doing that, you remove bias from your
Yes.
experimental design. It's a really simple thing to do, something that pharmacologists perhaps have been a little bit slow to adopt, but it doesn't take much time to do it, and it's an easy addition to your experimental process that actually will make your data output much more robust at the end. I realize that we are actually at time. So I'm assuming, I don't know, actually, Mara or Gabriele, if there are any further questions, will they be shared with us via email, and we can reply to them just in case anyone has any other questions that they might like to pose?
Yes, they can also put on the virtual stand you have from BJP. They can comment over there if they still have questions, just an option. This activity was recorded and is going to be presented on the virtual stand as well. So if anyone lost anything, you can go over there.
Wonderful. I know that you've got other things happening at the meeting, and so I should probably bring this to a close. Thank you very much, Maro and Claire, for speaking in this session, and thank you to all of those who've attended. Please continue to post your questions via the virtual stand or here, and we will get back to you.
Thank you. Goodbye.
Thank you. Bye-bye.